Namaskar दोस्तों, welcome to channel Toxicology on Board. Today in this video we'll be discussing another DABT topic, that is actinomycin B, its biological activity. Mainly we'll be going through the mechanism of action of this particular antibiotic. Let's directly dive into the question. This question is taken from ABT recertification exam, May 12, 2000. So the question was, the biological activity of actinomycin D is due to its. So we need to identify the mechanism how this actinomycin B is acting. So we had option number A, that is capacity to intercalate with DNA. Option B is damage to plasma membrane. Option C is incorporation into the RNA. Option D is impairment of cellular transport, ability to cause mitochondrial dysfunction. These are the various ways through which a antibiotic will show its biological activity. So the right answer will be in the green text, that is the option A, capacity to intercalate with DNA. So actinomycin D, it will go and intercalate with DNA and it shows its biological activity. I would request you to go back and look for other examples. Like for example, damage to plasma membrane is there. What is that antibiotic or any other drug which is doing, which is showing its biological activity by this method? Also, incorporation into RNA, impairment of cellular transport. Is there any drug which is doing this kind of mechanism, and ability to cause mitochondrial dysfunction? So that you will be understanding examples, you will be able to remember. Okay. So any example with similar kind of question, instead of capacity to intercalate with DNA, other options are there. You will be able to answer in the exam. Okay. So let's in this video we'll try to read in detail, understand how this actinomycin B is acting, showing its biological activity. So we have the explanation which is given is, the biological activity of this actinomycin D is due to its ability to bind tightly to the double stranded DNA. Okay, you have double helix DNA will be there, no, to which this actinomycin D will go and bind, and as a result it will inhibit the essential cellular processes. What are those? Primarily transcription, that is RNA synthesis. It will stop. Okay, so how this is means, understanding its specific binding site and mechanism of action is very important. Where certain drug is going and binding which part of the DNA. So in this case, actinomycin D, if you see, then only we'll be able to uh, play with that molecule. I mean to say, we can use it, design it for the chemotherapy or anti-cancer treatment, particularly for treating this type of cancer, that is Wilms tumor, rhabdomyolysis, and Ewing's sarcoma. Okay, so this is your actinomycin D, this is phenoxazone ring, and this is your DNA. Okay, intercalating DNA. So this is your, you can see the blue color part is, you know, this is your drug which will go and intercalate with the DNA. Okay, and you will also come across this important point, will come across RNA polymerase 1 will be there. Actinomycin D will go and bind with this topoisomerase 1 and 2. Okay, actinomycin D, it will inhibit DNA repair and induce autophagy. Okay, try to remember this pointer. So this is the typical structure of actinomycin D. Okay, so how the actinomycin D works means, one is DNA intercalation. The planar phenoxazone ring, this is your phenoxazone ring of actinomycin D, it will insert itself into the minor groove of your DNA double helix will be there, no? Strong preference over guanine cytosine GPC sequences. As you know, which particular part of the DNA helix it will, which where means GPC, that is guanine cytosine GPC sequences. Then coming to stabilization, the two cyclic pentapeptide, these two things are there, no? Side chains attached to this phenoxazone ring is there, no? It will fit snugly into the minor groove, whatever the DNA helix minor groove there, no? In that, forming hydrogen bonds with the guanine residues and other hydrophobic interactions with the surrounding nucleotides. Okay, this will anchor the drug firmly into the DNA. Okay, so if you look into the mechanism of inhibition, the formation of this stable DNA and drug complex is formed. Okay, it will prevent the DNA from further unwinding. So there will be no separation of this your double stranded helix. No, there is unwinding, it has been stopped. The physical obstruction effectively blocks the movement of the enzyme RNA polymerase. When you, in the first slide also you have seen, no, it will block the movement of this enzyme RNA polymerase along the DNA template, thereby inhibiting the elongation of RNA chains and stopping the transcription. RNA transcription is inhibited. Okay, So if you look into the secondary effects of this actinomycin D at higher concentrations, what this actinomycin D is doing, it can also interfere with the DNA replication. Primary thing is to stop this RNA transcription. Second one is also it can interfere with DNA replication and induce DNA strand breaks. Okay, within the DNA strands there will be breaks in the DNA takes place by interacting with the topoisomerase 1 and topoisomerase 2 enzymes. These mechanisms lead to cell cycle arrest. Okay, you have different cycles G1, S1, G2 the phases will be cell cycle arrest takes place and ultimately programmed cell death. Apoptosis will happen, which accounts for use as this is the thing which is based on which it is used. This particular actinomycin D is used as an anti-cancer drug. Okay, so if you this we have clearly seen that this is the actinomycin D. This is the phenoxazone and two side chains are there. Okay, and if you see this is your DNA and in which your drug will go and groove inside it. Okay, crystallized AMD with short DNA and proved AMD binding mode, that is intercalation will takes place. Okay, particularly that GAA, GCTTC AMD as observed in the crystal structure. Okay, and this is the structure of the DNA and AMD complex. DNA and actinomycin D complex determined by X-ray diffraction method. So under X-ray diffraction method, these are the pictures you can see. Anti-cancer drug actinomycin D binds to the DNA. This is the blue color thing is there. No, this is your drug. In the groove it this actinomycin D will go and bind, and here you can clearly see this actinomycin D. It will stop this topoisomerase 1 as well as topoisomerase 2 DNA replication, and because of which further unwinding of these two strands of the DNA will be stopped. Okay, so hope from this video you were able to learn about actinomycin D, the biological activity. As I told you, also read about further examples also, whatever which are given in the options, so that any question with respect to that one, you'll definitely able to answer in the exam. Thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel, Toxicology on Board. Thank you so much. All the best for your exam.